Hi, this is Tyler Foster, and I'm here to bring you to the finish line in the last section of biochemistry, the metabolism section. Metabolism encompasses the various processes that contribute to a cell's energy requirements and also waste removal. This involves the creation and interconversion between the four different classes of biomolecules, the carbohydrates, lipids, proteins, and nucleic acids. Each person learns differently, but I do encourage you to grab a notebook and draw the pathways as I cover them. Through our metabolic journey, I'll try and emphasize the frequently tested enzymes and concepts to get you maximum points for test day. Let's go ahead and begin. It can be useful to group metabolic reactions by the site in which they occur. Most metabolic reactions occur in either the mitochondria of a cell or the cytoplasm, although some reactions do occur in both. Don't be surprised if a USMLE question asks where a certain reaction takes place, so make sure you know these well for test day. In the mitochondria of our cell, reactions such as fatty acid oxidation, also known as beta oxidation, acetyl-CoA production, the TCA cycle, also known as the Krebs cycle or the citric acid cycle, our oxidative phosphorylation, and our ketogenesis takes place. In the cytoplasm, our cytoplasm is going to play host to our glycolysis, fatty acid synthesis, the HMP shunt, protein synthesis in the rough endoplasmic reticulum, steroid synthesis in the smooth endoplasmic reticulum, and also cholesterol synthesis. Now, protein synthesis can also take place on the rough endoplasmic reticulum. Do you remember what types of proteins are produced on our rough endoplasmic reticulum? Well, we have three main ones. Proteins that are meant to be secreted from the cell, proteins that are meant to be bound within organelles, or proteins that are embedded in the membrane are all going to be synthesized on the rough endoplasmic reticulum. On the other hand, our steroid and cholesterol synthesis occur on our smooth endoplasmic reticulum. Now, what reactions take place in both the mitochondria of the cell and in the cytoplasm? Remember I said that some do play a role in both. These are going to include the heme synthesis, the urea cycle, and gluconeogenesis. An easy way to remember this is that it takes two to hug. So here's our two cats hugging. H stands for heme, U stands for urea, and G stands for gluconeogenesis. Now, by the way, do you remember what vitamin is required for heme synthesis? That would be the vitamin B6, pyroxidine, which is used to convert the succinyl-CoA and glycine into aminolevulinic acid, the first step in making heme. Let's see how well you're paying attention. Glycolysis occurs in either the mitochondria or the cytoplasm, whereas the Krebs cycle, the TCA cycle, or the citric acid cycle, is going to occur in which one? Well, it's important to know where these two paths occur. Glycolysis is going to occur in our cytoplasm of the cell, whereas the TCA cycle or the Krebs cycle is going to take place within the mitochondria. This will be an easy test point, so make sure you know where these reactions are taking place. Before I introduce a bunch of specific enzymes, I do find it useful to learn some terminology first. The name and the function of an enzyme are most often related. If you can understand the naming terminology, you can figure out what the basic function of that enzyme is. Let's briefly cover some of the more important enzyme categories. We'll start off first with a category of enzymes called the kinases. Enzymes that are kinases are going to transfer a high energy phosphate group from ATP to a target substrate. An example of this would be the phosphofructokinase enzyme. So you'll find phosphofructokinase in the glycolysis pathway, or remember how glucose is broken down to create our ATP. Phosphofructokinase is going to phosphorylate, or add another phosphate group, to our fructose 6-phosphate molecule in glycolysis. This is going to create a molecule of fructose 1,6-bisphosphate. Notice how the phosphate group is going to be taken from our ATP molecule and thus moved onto our fructose 6-phosphate molecule. So kinases are simply going to remove a phosphate group from ATP and add it to a substrate. Now, what do you predict a phosphorylase enzyme does? Well, our phosphorylase enzymes are similar to their kinases, which we just talked about, meaning they add a high energy phosphate group onto a substrate, but instead, our phosphorylase enzymes are going to do it without using ATP. An example of a phosphorylase would be the glycogen phosphorylase, which is going to be found in our glycogenolysis pathway. Glycogenolysis is where you take a large glycogen chain and then break it off to create free glucose molecules. Now, our enzyme glycogen phosphorylase is going to grab a free phosphate and cleave the 1,4 glycosidic bonds in glycogen to produce our free glucose molecule, our glucose 1 phosphate, by adding this inorganic phosphate to it. So here we take our glycogen chain, we're going to grab a free phosphate molecule using our enzyme, which is a phosphorylase. We're going to transfer that phosphate onto our substrate, creating a molecule of glucose 1 phosphate. Our next set of enzymes, the phosphatase enzymes, are going to be the opposite of our kinase and our phosphorylases, which we just talked about. The phosphatases, like fructose 1,6-bisphosphatase, are going to remove phosphate groups from its substrate. Fructose 1,6-bisphosphatase is going to convert our molecule of fructose 1,6-bisphosphate into a molecule of fructose 6-phosphate. So here we saw a substrate with two phosphate groups. After we undergo enzymatic reaction, we're left with a product that only has one phosphate group attached. So the phosphatases are going to cleave off a phosphate group. As a side note, we look at the numbering system. There's a fructose 1,6-bisphosphate. That 1 and the 6 means that there's a phosphate group attached at the 1 carbon and at the 6 carbon. And over here, since we only have one phosphate group attached, we only have a phosphate group attached to the 6 carbon. The group of enzymes that are our dehydrogenase enzymes are simply going to catalyze oxidation reduction reactions. Now you've heard that term before, oxidation reduction reactions. Do you remember what a redox reaction is? That means that hydrogens, or electrons, are being removed from one substrate and then added to another. So an important enzyme that we're going to cover is going to be our pyruvate dehydrogenase, which converts a molecule of pyruvate into our acetyl-CoA, which can then go on to our Krebs cycle. Now here we see pyruvate dehydrogenase moving electrons and protons to create NADH molecules, which will then be used for later use. Looking at this enzyme, pyruvate carboxylase, which is going to be our carboxylase enzyme, this is going to move a CO2 group onto a substrate, as you can see here. A normal pyruvate molecule has a CO2 group attached to it, and after, with the help of our pyruvate carboxylase, we're going to add another CO2 group to form oxaloacetate, which is going to have two CO2 groups. So remember, our carboxylase enzymes are going to add a CO2 group. Now, the carboxylase enzymes need the help of biotin, aka vitamin B7. Now, whenever you hear the word carboxylase, I want you to automatically think biotin. On that last slide, we talked about the carboxylase enzymes. So what would you call the set of enzymes that remove CO2 groups? Well, hopefully you're not thinking too hard. Removing CO2 groups is the duty of our decarboxylase enzymes. So here we have a CO2 group, and magically it disappears. Enzymes that add our hydroxyl group, an OH group, are going to be called the hydroxylase enzymes. And then lastly, we have our mutases, which are a set of enzymes that move a functional group within a molecule. An example of a mutase would be methylmalonyl-CoA mutase. Understanding the naming of an enzyme will help in the understanding of the different metabolic pathways in the body. This whole biochemistry metabolism section is just explaining the process of how molecules get enzymatically altered to make our new molecules. We'll see that we have many different processes at our disposal, and when one of these has a dysfunction, then we get different diseases. It's also important to know the rate determining enzymes for the various metabolic processes. These are the most commonly tested enzymes since they determine the rate at which these reactions can occur. These rate determining enzymes are also the ones that are most commonly targeted by drugs because slowing these down or speeding them up will have an effect on these processes that they're involved in, which might not be the case for the other enzymes in the pathway. It'll definitely help you on your test to know which enzyme is rate limiting for each process and how that enzyme is regulated. For example, let's go ahead and talk about HMG-CoA reductase from down here. HMG-CoA reductase is the rate-limiting step of cholesterol synthesis. HMG-CoA reductase is going to convert a molecule of HMG-CoA into mevalonate, which is then eventually going to go on to become cholesterol. So, do you remember which class of drugs inhibits the enzyme HMG-CoA reductase? Well, that would be our statin drugs, which are used to treat hypercholesterolemia. The statin drugs are going to inhibit the rate-limiting step for cholesterol synthesis here. So we have our statins and it's going to cancel out that enzyme, thus we're not going to produce as much cholesterol. Now we'll cover all of these throughout our lecture, so I'm not going to read them off to you now, but you might want to come back to this and review it later, maybe using flashcards. I'll discuss how each enzyme is regulated in detail throughout this chapter. If you're following along in the first aid book, this summary of pathways diagram may be a little bit intimidating at first, but it's meant to show you how all the different metabolic pathways we're about to go over are interrelated to each other. I simplified it here on the slide just to make it a little bit easier to interpret at first glance. The take-home message from this is that carbohydrate metabolism is connected to protein metabolism, to lipid metabolism, and even nucleic acid synthesis. In this figure, just recognize how everything is interrelated within the body. We'll talk about each of these processes throughout our lecture today, and then once you feel comfortable with the various smaller pathways, it'll be helpful to look back here and to see how it all fits together in this nice big picture. We'll slowly introduce enzymes, substrates, and products that will fill this box diagram into a more complete picture. This is also a great page in your book to take notes from other parts of the biochemistry chapter or from other chapters as well, since there's a lot of inputs and outputs to this diagram that are not shown here. For example, 1,3-BPG is also used to make 2,3-BPG. So this 2,3-BPG is going to be used in erythrocytes to reduce the affinity of hemoglobin to oxygen, which will be talked about more in the Hem/Onc chapter. Also, fatty acids are broken down into acyl-CoA, which is transported using the carnitine shuttle to the mitochondria, where they'll be converted into acetyl-CoA, which can then enter the TCA cycle. So as you study, you'll probably find lots more things to add to this page. For now, don't feel like you need to memorize all this before we move on. Just realize that this diagram in the book should make a little bit more sense after we break all these different pathways down. We'll start to deconstruct that elaborate summary of pathways diagram by first talking about energy production. What is the main energy molecule the body needs to survive? Well, hopefully you've heard about adenosine triphosphate or ATP before, which stores its energy via these high energy phosphate bonds. The body has multiple processes to create ATP depending on whether oxygen is present or not. When oxygen is present, it's called aerobic metabolism, and when oxygen is absent, it's called anaerobic metabolism. Aerobic metabolism takes place in both the cytoplasm and the mitochondria, where oxygen is the final electron acceptor. Aerobic metabolism of glucose can produce 32 molecules of ATP via the malate aspartate shuttle, which is found in the liver and the heart. This shuttle transports NADH electrons gathered in the cytoplasm to the mitochondrial NADH acceptor, which can produce 3 molecules of ATP per molecule of NADH. Only 30 molecules of ATP are produced when the glycerol 3-phosphate shuttle is used in the muscle. So now we're in the muscle. The difference in this shuttle is that it transports NADH electrons from the cytoplasm to the mitochondrial FADH2 acceptor, which can only produce 2 molecules of ATP per NADH consumed. Anaerobic ATP production takes place in the cytoplasm of the cell, and it does not require oxygen. It produces 4 ATP total, but it requires 2 ATP to be invested, so actually anaerobic metabolism has a net gain of 2 ATP molecules per molecule of glucose. ATP can also be manipulated and hydrolyzed in otherwise energetically unfavorable reactions that would actually not occur spontaneously to provide the necessary reactant for a reaction to occur. Now remember, I said that each process in the body has a certain function. For example, the function of glycolysis, the Krebs cycle, and the electron transport chain is to produce large amounts of ATP for the body to use. Arsenic poisoning disrupts glycolysis, so arsenic will actually inhibit the body's ability to create ATP. And when a cell is starved for ATP or energy, it'll actually die. So we should try our hand at a flash quiz. A 25-year-old runner is exercising to the point of requiring anaerobic glycolysis. How many ATP molecules per glucose molecule is she going to produce at this point? Well, in the case of anaerobic metabolism, only a net of 2 ATP are produced per molecule of glucose. So this is dramatically less than that 32 ATP produced by the malate aspartate shuttle when oxygen is present or available for aerobic metabolism to occur.